Yes, students, let me start with a new chapter, that is sexual reproduction in flowering plants. Okay. Now, the first word that you're supposed to know is flower. So, what is a flower? Flower is said to be a modified vegetative shoot. Vegetative shoot meant for sexual reproduction. Flower is a modified vegetative shoot meant for sexual reproduction. Now, when I use the word modification, please remember any change from the normal structure, function or position of plant or its parts is said to be modification. We can't use the modification in case of animals or human. It becomes handicapped. But here it is only for plants that we use the word modification. So what am I using here? Flower is a modified vegetative shoot. Then how should a vegetative shoot be and what has happened to the vegetative shoot so as to form a flower? Let us see. Okay. Now this is the stem. The thick regions of the stem is said to be node. It is at the node the leaf arises. In the axil of a leaf, a bud arises. This bud is said to be axillary bud. The bud is said to be axillary bud. Depending on the type of plant, this axillary bud may give rise to, okay, depending on the type of plant, this bud may give rise to a single flower. Please remember, single flower or cluster of flowers called inflorescence, or it may give rise to a branch. Let us assume that this axillary bud gave rise to a branch with 4 nodes, 1, 2, 3, 4. Evolutionary study says that the entire branch was compressed and gave rise to 4 whorls of a flower. That is, I told you 4 nodes. Each node gave rise to 1 whorl. The units were of different types, brightly colored and showed special functions. Thereby we said it is a modified vegetative shoot. So, this stem or a branch is said to be a vegetative part of a plant, isn't it? Thereby we say this branch was modified. Hence the name flower is a modified vegetative shoot. And later it showed the function of sexual reproduction. Is it clear? So, 1, this is the first one, this is the second one, third and fourth. These were modified into 4 whorls. Which are the 4 whorls of a flower? The first whorl is said to be calyx. Second whorl, corolla. Third whorl, andrecium. And the fourth whorl, gynoecium. So, which are the 4 whorls of a flower? Calyx, corolla, andrecium and gynoecium. The first whorl, that is calyx, appears to be green in color. Its individual units are called sepals. What is the individual unit of calyx called? Sepal. Thereby we can say collection of sepals is calyx. So also we have corolla. It is brightly colored and the units of corolla are said to be petals. So what are the individual units of corolla called? Petals. In other words, we can say collection of petals forms the second whorl, that is corolla. Okay. What is the function of calyx? It protects the bud or other parts in young condition. It protects the flower when it is in bud condition, I can say. Whereas petals are brightly colored, I told you. They attract the insects and helps in pollination. But remember, these two do not take part in sexual reproduction. In the sense, they are not necessary for the formation of fruit. Hence, we say these are non-essential whorls of a flower, non-essential whorls of a flower, or they are said to be accessory whorls of a flower. They are said to be accessory whorls of a flower. On the other hand, which is the third one? Androe, you have to remember, andrecium. This is the male reproductive organ of a flower. This is the male reproductive organ of a flower. It also has many units. So, individual units of andrecium is said to be stamen. On the other hand, we have the fourth one, that is gynoecium, gynoecium. This is the female reproductive organ of a flower. Female reproductive organ of a flower. Its individual units are said to be carpel. Thus, what are the individual units of each whorl? Calyx, sepal, corolla, petal, andrecium, stamen, and gynoecium, carpel. Okay. Individual unit of gynoecium is said to be carpel. Let us see the other details of it. Before that, please remember, flower is said to be a fascinating organ because it forms the object of ornamental, religious, or even it has cultural value. In addition, it is said that it is used in conveying important feelings of human like love, affection, happiness, etc. Thereby, flower is said to be a fascinating organ. Is it clear? Okay. Let us see the details of calyx, corolla, andrecium and gynoecium in detail. Before that, let me tell you that andrecium and gynoecium are said to be the essential whorls of a flower. They are said to be essential whorls of a flower because they take part in sexual reproduction. They are necessary for the formation of a fruit. Okay? Yes. What is thalamus? It is the condensed axis of a flower. It is from the thalamus all the four whorls of the flower arise. Now, when I say condensed axis and the four whorls arise, the units are not seen only on one side. They are seen around. Okay. Thereby, we use the word whorls. It doesn't mean that you can see on one side and not on the other side. However you turn the flower, you can see the units around. So, how is it diagrammatically represented? It is diagrammatically represented in the form of circles. But remember, innermost whorl is gynoecium. Okay. And outermost whorl is calyx. We can't start from calyx and then proceed inwards. You cannot draw neatly. Thereby, I will just show you how to draw. Start from the gynoecium. Followed by the gynoecium is the andrecium. And then this is the corolla. And this is the calyx. Okay. This is the first whorl. Many a times I have told you. This is the second whorl. This is the third whorl, andrecium. And this is the fourth whorl, gynoecium. Suppose they ask you in the exam, mention the whorls of a flower, then you are supposed to write all the four whorls like calyx, corolla, andrecium and gynoecium. Instead, if they ask you, mention the non-essential whorls of a flower, then you have to write is only calyx and corolla. In addition, you will have a question, mention the first non-essential whorl of a flower, then you have to write calyx. Second non-essential whorl of a flower is corolla. In the same manner, these two, that is andrecium and gynoecium, are said to be the essential whorl of a flower. Suppose I ask you mention the essential whorl of a flower, you should write andrecium and gynoecium. Instead, they ask you mention the second essential whorl of a flower, then you have to say gynoecium. first essential whorl of a flower, andrecium. Okay. So, this is first essential, this is second essential, this is first non-essential, and this is second non-essential. It is very simple. Please be alert while reading the question. Read the question twice, answer once. That means what? You should read the question properly, whether it is essential or non-essential, just whorl, okay? Or is it the first non-essential, second essential, only the whorls, etc. Okay, read the question twice. Then you need not repent later for knowing the answer and not representing. Is it clear? So, this is about the whorls of a flower. Suppose I take the LS of a flower, how does it appear? Let us see. See, the calyx appears like this. This is the individual unit of calyx, which is called sepal. These are the petals. Okay. So, individual unit of corolla is called petal, and then comes the andrecium. These are nothing but the stamens. These are the anthers of stamens. What is a stamen? I told you individual unit of andrecium is called stamen. Okay. What about gynoecium? I'll show you. This is the gynoecium that is present inside. This is the stigma. Okay. What is this now? It is nothing but gynoecium. You can see now. Why did I draw LS? To make you understand that whatever we see at the top is not the first whorl. On what basis do we say it's the first whorl? If you see the base of that unit, you can see first the calyx, okay? The calyx appears like this completely. When you remove this calyx, you can see the base of the corolla. Okay, you can see the base of the corolla. On removing corolla, you can see the base of the andrecium. This is the base of the andrecium. On removing andrecium, you can see the base of the gynoecium. That is, it appears like this. You can see the base of the gynoecium. Thereby, how do you say that it is the first whorl, second whorl, etc.? It is only based on observing the base of that particular whorl. Okay. What about the other details? What are the parts of a stamen? Let me tell you. When I told you about calyx, it is only the sepal and only the petal with reference to corolla. But what about the stamen? Stamen is the individual unit of andrecium. It has a long stalk-like structure called filament and a bag-like structure called anther, which in future produces pollen grains. And this is the connective which connects the filament to the anther. Okay. Now, this is the thalamus from where the filament arises. Please remember, it's a common question. The part of the filament that is attached to or that arises from the thalamus is said to be the proximal part of the filament. Proximal part of the filament. Whereas the part of the filament that is attached to anther, okay, this region is said to be distal part of the filament. So, which is the part of the filament that is attached to the thalamus? It is the proximal part that is attached to the thalamus. Is it clear? Okay. These are the parts of what? They are the parts of a stamen. Let us pass on to the parts of a carpel. So, all of us know that individual unit of gynecium is said to be carpel. So, what are the parts of a carpel? It is ovary, style and stigma. Okay, this is the swollen portion, that is ovary. This is the long stalk-like structure, that is style, and this is stigma, the landing place for pollen grain. Is it clear? Finally, what is the individual unit of androecium? Stamen. What are the parts of a stamen? Filament and the connective. And on the other hand, what is a gynecium? Female reproductive organ of a flower. Its individual unit is said to be carpel. What are the parts of a carpel? Ovary, style and stigma. Is it clear? Okay. Let us see the other details of androecium and gynecium. This is what we say or explain under the heading pre-fertilization changes. That means if a flower is to be ready for fertilization, what hormonal changes should it undergo? That is what the structures, the hormonal changes so as to form pollen grains, male gametes, and then in case of female, the egg or female gametophyte, etc. Everything should be ready just before fertilization. That is what we say pre-fertilization events. That is what we say pre-fertilization events and structures. Okay, or first we study the structure and then the event. Yes, students, just few minutes back I told you about androecium. Its individual unit is called stamen, and the parts of a stamen are filament, anther and connective. It's a tissue that connects the filament and the anther. Now let us see the details of it. Anther is a bag or a sac-like structure which in future produces pollen grains. Now this anther may be of a single lobe wherein we call it as monothecous anther. Monothecous anther. Even if it is single lobe, it has two spherical structures in it. These are called microsporangia. Microsporangia, they are circular, okay, they are circular bag-like structure. Now on the other hand, the anther may be bilobed. In that case, we call it as dithecous anther. Okay, if it has two lobes. Anther with single lobe is monothecous anther. Anther with two lobes is said to be dithecous anther. I told you, if it is a single lobe too, it will have two microsporangia. So thus now you can imagine that these are the four microsporangia because it is dithecous. Each bag-like structure has two microsporangia. Finally, we find here we say two microsporangia. Here we say four microsporangia, or we also call it as tetrasporangiate. Sporangiate. Okay, we call this as what? Tetrasporangiate. What does the LS of anther show? It is a slanting LS that we show here. See, it appears like this. And we find the microsporangia. Okay. Please remember, these microsporangia later at maturity join to form pollen sacs. When after the formation of pollen grains, they join to form pollen sacs. That is, in each lobe there are two. So, two of them join to form pollen sac. One pollen sac here, one pollen sac. Thereby, I said two pollen sacs. Okay. Totally, we say it is two pollen sacs. Yes, now. How does microsporangia appear and what it is? Each microsporangium, single is microsporangium, plural is microsporangia. Each microsporangium is a circular bag-like structure covered by 4 walls. The innermost wall is said to be tapetum. The innermost wall is said to be tapetum. It is rich in cytoplasm, or I can say it possesses dense cytoplasm and multinuclei. So it is multinucleated with dense cytoplasm. Okay. Next to this, we see 2 to 3 layers of cells. Okay. Just start from the center, mark tiny vertical lines, and then join this. Okay. This is how we have to draw. Please remember to have a rough sheet, a rough book, paper, pencil, eraser, etc. Whenever I draw the diagram, try to draw it. If not, soon after I finish, too, you can draw looking at the technique. Is it clear? Not listening to the lesson today and some other day. If you go to practice, you can never do that. The same day you have to practice, the same day you have to listen, and it is the same day you have to try to answer the questions that is given below. Okay. Please be ready with your PDF too, because soon after I explain, I'll just mark the important points that already I have explained. Okay, that I have already explained. I'll be marking for you and telling you which are the 1 mark question, 2 marks questions, etc. You can even have a marker. That will be best. Okay. Yes, this is 1 layer. I told you 2 to 3 layers. Let us show 2 layers. Now, followed by this, we see 1 layer of cell, a single layer of cell. And then outermost, we see 1 layer of cells. Yes, this is said to be epidermis. The second layer, okay, the second one is said to be endothelium. Third, I told you 1 or 2 layers. These are said to be middle layers. They are not 1, I told you 2 or 3. And finally, we have the innermost layer, which is said to be tapetum. Now, what is this circular structure? It is the microsporangium, single. It encloses number of cells. All the cells are almost uniform, thereby they are called sporogenous cells, or they are said to be microspore mother cells. Okay, what are these called? They are called sporogenous cells or microspore mother cells. Or even it is said to be pollen mother cells, because in future it produces pollen grains. It is called microspore mother cells because at first it produces microspores. Okay, this is a single microsporangium. It is the section of a microsporangium. Let me show you the enlarged diagram of this TS. Okay, this one. Have a look at this here. I am marking. How does it appear? It shows 2 bag-like structures, Now the two bag-like structures like this. This is the TS of a Dithecus anther showing 4 microsporangia. This is of one lobe, these two, okay. These two are of one lobe, these two are of another lobe. So this is TS of Dithecus anther. So this is the microsporangium, okay. This is the microsporangium, and these are the wall layers. So that is what I have shown you, one microsporangium. Is it clear? Okay, students, let me explain once more. This is the microsporangium. It is covered by 4 wall layers. The first is the epidermis, second is the endothecium, third the middle layers, and the fourth is the tapetum. So the first 3 layers helps in protection and dehiscence of anther during the release of the pollen grain. Okay. Only when the anther dehisces, the pollen grains will be released. When will the pollen grain be released? Only when it is matured. So in other words, I can say, after the pollen grains are matured, the first 3 layers, namely epidermis, endothecium and middle layers, start showing dehydration, and thereby it ruptures or dehisces to release the pollen grains that are formed in the microsporangium. In the beginning, the microsporangium has the young cells, that is the sporogenous cells or the microspore mother cells, which are to be matured to produce pollen grains. Okay. So the first 3 helps in what? Protection and dehiscence of anther. What about this tapetum? Tapetum is the innermost layer of the wall of the microsporangium. That is, it has dense cytoplasm and a prominent nucleus. It supplies, okay, prominent nuclei, not one, many. It supplies nourishment to the developing sporogenous cells. Because of this nourishment only, finally we see the formation of microspores, and later they mature to form pollen grains. Is it clear? Yes. Before that, I just want to tell you, this is the TS of the Dithecus anther. If you just consider this completely, if you consider it is the LS, I will just show you the TS, how does it appear. This is one lobe, this is another lobe, okay, or this is the first lobe, this is the second lobe. The other part of the first lobe is here, this is one. So this is one lobe, first lobe, this is the second lobe. Thereby we say Dithecus anther. One microsporangia of first lobe, another microsporangia of first lobe. Then here microsporangia of second lobe, microsporangia of second lobe. Okay. These are the wall layers. This is the microsporangium. So I have shown you a portion, that is a section of this. Okay. One portion enlarged. I have shown you the details. Is it clear? Yes. Let us pass on to the next topic, that is microsporogenesis. The name itself says microspores are genesis, formed. So how is it formed? Let us see. Is it clear? Okay. Yes, students. Let us see what is microsporogenesis, and then structure of a pollen grain, and in addition development of pollen grain to male gametophyte. All 3 topics are very important and are linked to one another. Please have a look. At first, what should I explain? Yes, let me start with microsporogenesis. So this is the microsporangium. Just few minutes back I have explained, isn't it? These are the cells which are homogeneous, and we call them as what? Microspore mother cells, or we say they are the sporogenous cells. Okay. These cells, that is the sporogenous cells, will act as the microspore mother cells. Each cell of the sporogenous tissue Okay, what is this totally called? Sporogenous tissue. In a microsporangium, each cell of sporogenous tissue acts as what? A potential microspore mother cell. It acts as a microspore mother cell. Thereby what happens to it? Let us see. Now let me assume this as one microspore mother cell. Microspore mother cell, right? This is diploid in nature. This microspore mother cell undergoes meiosis so as to form 4 haploid cells which are attached. They are all haploid. After meiosis, we see haploid cells. They are 4 in number. It is said to be. It is said to be what? It is said to be pollen tetrad or microspore tetrad. Each microspore mother cell, which is diploid, undergoes reduction, division or meiosis so as to form 4 haploid cells which are still attached called pollen tetrad or microspore tetrad. Later, they separate to form pollen grains when at maturity. When the anther wall gets dehydrated and is about to release the pollen grains, okay, it gets separated. So these are said to be the pollen grains. When they are still attached, I told you it's microspore or pollen tetrad. Now I can call this as pollen grains. Meanwhile, each pollen grain will have attained a certain structure. So what is that structure? Let us see the structure of a pollen grain. Each pollen grain is spherical in shape, enclosing dense cytoplasm and prominent nucleus. So if this is the nucleus, okay, if this is the nucleus and this is the cytoplasm, what should this be? This outer wall should be the plasma membrane. So this is the plasma membrane. This plasma membrane is surrounded by 2 layers or 2 walls. The first layer that is close to the plasma membrane is smooth and it is called intine. It is made of cellulose and pectin. What about the next layer? Followed by this or outer to the inner layer is the presence of a layer which is called exine. So this layer is said to be exine. This is the outer layer, this is the inner layer, okay, and 2 together forms a covering for the pollen grain. This outer layer or exine is spiny, okay, it is spiny or elongated, or it is variously sculptured, variously designed, or we say variously sculptured. Just by trapping the pollen grains and observing under the microscope, it is possible for us to detect to which plant this pollen grain belongs to. Number of pollen grains will be there in the air, isn't it? Once we trap that and observe under the microscope, by looking at the exine and its sculpture, it is possible for us to decide, or we will come to know to which plant it belongs to. Okay. So exine is made of a special resistant organic material or chemical which is called sporopollenin. Whereas intine is made of cellulose and pectin, and finally we have plasma membrane, cytoplasm and nucleus. So, this is the structure of a pollen grain, and please remember it is haploid because the microspore mother cells has already undergone meiosis. Is it clear? Let us see the details of the sporopollenin and the other details. Yes students, before passing on to the other details of pollen grains, let me just recall for you, okay, microsporogenesis. It is the process of formation of microspores from microspore mother cell by meiosis. Okay, it is a process of what? Process of formation of microspores from microspore mother cells by meiosis. Thereby, the microspore mother cells should be diploid because it is undergoing meiosis, and hence microspores will be haploid. Okay. Later they mature to form pollen grains. That is what I told you, the structure of the pollen grain, that is the exine, intine, plasma membrane, nucleus and cytoplasm. Hope you remember sporopollenin. This sporopollenin is a special organic material that is resistant to what? It is resistant to strong acids. It is resistant to strong alkalis. Then it can withstand high temperature. And it can withstand any adverse climatic conditions. Okay, sporopollenin can withstand any sort of adverse climatic condition. So, this is present where? In the exine of pollen grain. Throughout it is present, except, I will just erase and show you, except leaving small openings here and there. That means these are the regions which are devoid of sporopollenin. Sporopollenin. What are these called? These are called germ pores. What are the regions on the exine devoid of sporopollenin called? They are called germ pores. The name itself says it helps in the germination of the pollen grain. How? Let us see. Before that, I will just repeat once more. Organic material that can withstand strong acids, alkalis, temperature, adverse climatic condition. It is said that it will not degrade for any of the enzymes too. None of the enzymes can also degrade it. Okay. Hence, pollen grains can be stored for many years in the form of fossils. They will ask you the reason. How is that pollen grains can be stored in the form of fossils for years together? That is because of the presence of what? Sporopollenin. It's a common 2 marks question. What's the function or significance of sporopollenin? So, germ pores also I told you. Yes, please have a look now. If this is the anther which is about to dehisce, half and half I have shown. These are the pollen grains. Okay. Here I have shown single cell. What happens at maturity? When it is ready for the dehiscence. During that stage, what happens to each pollen grain is, each pollen grain which is haploid and single cell will undergo mitotic division. The cells do not separate, they undergo mitotic division, and moreover it is not equational division. When I say mitosis, it is equational division. But what happens here? Unequal division occurs such that one of the cell is small and it is called the generative cell, and the other cell is large, which is called vegetative cell. Okay, this is said to be the vegetative cell. The large cell is said to be what? Vegetative cell. So, the vegetative cell and generative cell. So, now what do you call this pollen grain as? Two cell stage of pollen grain. So, at maturity in the angiosperms only, what do we see? We see the pollen grains to have been matured into two cell stage structure. So, once it is two cell stage in structure, it is ready for pollination. So, this is a common 1 mark question. What stage of pollen grain is ready for pollination? It is a two cell stage of pollen grain that is ready for pollination. Is it clear? So, two cell stage of pollen grain is ready for pollination. Please remember, this is the dehystanther wherein the pollen grains are coming out. Once the pollen grains comes out, it will be carried away by agents, and then when it falls on a suitable stigma, it brings about fertilization, before which the two cell stage pollen grain undergoes number of changes on the stigma. Are you understanding? As soon as the pollen grain lands on a receptive compatible stigma, okay, as soon as it lands on a compatible stigma, that means the stigma should be of mango. So, this pollen grain should also be of mango. That means they should be of the same species. Then only further development of pollen grain will take place. In other words, we say stigma receives the pollen grain. If not, if it is not of the same species, it rejects the pollen grain. So, what do you mean by accepting, receiving the pollen grain? This pollen grain will undergo further development on the stigma. So, two cell stage pollen grain becomes what? Let us see. Is it clear? Yes. Yes. Let me show you the details of the development of pollen grain into a male gametophyte. Okay. See, if this is the stigma, you learn in detail under pollen pistil interaction. This is a small explanation I am just giving you. This is the stigma. Okay. When the pollen grains of the same species falls on the stigma of a flower, that is a pollen grain, that is compatible. Pollen grain and the stigma should be compatible. That means they should be of the same species. Only then what happens to this pollen grain? Let us see. It shows further development on the stigma. That means what? A male gametophyte is formed. This is a pollen grain which is two cell. This is the generative cell. This is the vegetative cell. Okay, this is the generative cell. What's the other name for vegetative cell? It is said to be tube cell. What's the reason? In future, this vegetative cell forms a pollen tube, and this generative cell will give rise to two male gametes by undergoing again mitosis. Okay, these are the two male gametes that are formed from the generative cell by mitosis, and this is the pollen tube that is formed from what? From the vegetative cell. Hence, the other name for vegetative cell is tube cell. Now, this entire structure is said to be a male gametophyte. Okay. So, we say pollen grain represents male gametophyte. Only when it falls on a suitable or compatible stigma, Such a development takes place on the stigma itself, and further it will be sent inside to reach the ovary, and later the embryo sac. All those details you will learn under the heading pollen pistil interaction. Okay. After few classes you will come across this heading, that is pollen pistil interact. Yes students, please remember importance of pollen grains. Pollen grains are rich in nutrients. Thereby they are used as additional food supplement. Okay. Second, pollen grains can be stored for many years in liquid nitrogen, that is under minus 196 degrees centigrade. Okay. It can be preserved and used whenever we want, that is in the sense it is used in crop breeding program. Okay. These are the uses of pollen grain, that is rich in nutrients, used as a food supplement, either as in the form of tablets or any other form, and in addition it is stored for years together in the form of fossils, or we say under cryopreservation condition, wherein it can be used further. Okay. But on the other hand, when you consider the problems of pollen grain, we say they bring about allergic reactions. They bring about allergic reactions. They cause allergy, that is nose irritation, throat or even eye irritation, and sometimes the person may develop bronchitis or even asthma. That is what we say, pollen grains are very allergic to some persons, and very common pollen grain is the pollen grain of a plant, that is Parthenium. It is also called Congress grass. Okay. Now this Parthenium came as a, that means this plant came as a contaminant when it was imported along with the wheat plants. Okay. It came as a contaminant. Now it is spreading like anything, that is everywhere we see the pollen grains of Parthenium that causes allergy. But now to some extent they have seen to that it is being warded off. Yes students, let me pass on to the first chapter, that is sexual reproduction in flowering plants. See, having viewed the video, it will be easy for you if you go through the material. Okay. Once more I am repeating, first you should go through the video and then only take the material, have a pencil with you or a marker, and then make a note of it. Okay. I will just be marking 1 mark, 2 mark, 3 marks, 5 marks or even NEET questions for you. Is it clear? Yes, no. See, flower is a fascinating organ of angiosperms. Flowers are the objects of ornamental, religious and cultural value. They have been used as a symbol of conveying important human feelings like love, affection, happiness, grief, etc. Angiosperms are regarded as anthophytes. Anthos means flowers, phyta means plant. Flower bearing plants. Okay. On the other hand, flowers are the specialized reproductive structure or the modified vegetative shoot meant for sexual reproduction. Okay, passing on to the parts of a flower, these are very common 1 mark and NEET questions. Okay, in a flower there are 4 whorls, means if this is the axis of a flower which is called thalamus, the units of the flower are present around the thalamus, around, thereby we say whorl. Okay, then what is thalamus? It's a condensed axis of a flower. Okay, this is the condensed axis of a flower. Outermost whorl is said to be the calyx. Its individual unit is said to be sepal. Second whorl. Okay, these are the whorls of a flower. This is outermost whorl calyx, this is corolla, this is andrecium, and then gynoecium. Okay. So, the innermost whorl is the gynoecium, outermost whorl is the calyx. Now, when I say calyx, it appears like this. All the units together we say calyx, but one of it is said to be the sepal. So also corolla. Individual unit of calyx is sepal, individual unit of corolla is petal, individual unit of andrecium is stamen, and the individual unit of gynoecium is carpel. Okay. Calyx and corolla are said to be the essential whorls of a flower, whereas calyx and corolla are said to be the non-essential whorl of a flower. Calyx and corolla. Whereas andrecium, okay, andrecium and gynoecium are said to be the essential whorl of a flower. Okay. Why are they called non-essential whorl? Because they do not take part in sexual reproduction. Okay. They are said to be non-essential because they do not take part in sexual reproduction. See, these two non-essential or accessory. These to take part in sexual reproduction, thereby they are said to be essential whorls of a flower. Thus, calyx and corolla need not be present for the formation of a fruit, but andrecium, gynoecium must be present, okay, for the formation of a fruit. Sometimes we say that calyx and corolla are absent, but still fruits are formed. Suppose they are present, they help in pollination by attracting the insects. So, we see various adaptations among these four whorls. Okay. Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Okay, inflorescence is formed which bears the flower buds and then the flowers. So, what is an inflorescence? It is nothing but cluster of flowers arranged in a definite pattern on a reproductive shoot. Okay. So, we see cluster of flowers arranged together. So, this cluster of flowers arranged in a definite pattern is said to be inflorescence. Now, what is a stamen, microsporangium and pollen grain? Andrecium is the male reproductive organ of a flower and it consists of whorl of stamens. Individual unit of andrecium is said to be stamen. What are the parts of a stamen? Filament, anther, and then connective. Filament, anther, connective. This is a long stalk-like structure which attaches to the thalamus. This region is said to be proximal end of the filament. Whereas opposite to that is this end which attaches to the anther. This is called distal part of the filament. Thus, we see three parts: filament, anther and connective. Now, this is a sectional view. Okay. Now, let me explain further. Before that, just mark this. Long, slender stalk-like structure is filament. Bag-like structure is anther. In future, this anther produces pollen grain. Proximal end, just now I told you, and about the distal end. It's a bag-like structure which produces pollen grains at maturity. Yes, now have a look. The anther may be single-lobed or it may be bilobed. It may have single lobe or bilobe. Even though it is single lobed, it has two circular or cylindrical structures which are called microsporangium. Thus, in a dithecous anther, we find 1, 2, 3, 4 microsporangia. Okay. Single lobed anther is said to be monothecous anther, bilobed is said to be dithecous anther, and it is tetragonal structure showing 4 microsporangia, that is tetrasporangiate. Structure of microsporangium, this is also a very important question. A typical microsporangium appears circular and is surrounded by 4 wall layers: epidermis, endothecium, middle layers, and tapetum. Okay. Epidermis is the outermost layer, that is a single layer which is protective in nature. This one, it is protective in nature. Followed by that, we see the presence of endothecium, second layer, with thick cells which helps in dehiscence of anther and also in protection. Middle layers, 1 to 3 layers of cells are present here. Okay, that is what we say middle layers. They also help in dehiscence of anther and also they are protective in function. Thus, the first 3 layers helps in protection and dehiscence of anther, that means to release the pollen grains. It provides nourishment, whereas this is the most important one, favorite question for paper setters, tapetum, the 4th layer. It is the 4th and the innermost wall layer which has dense cytoplasm and multinuclei. It provides nourishment to the developing pollen grains in the microsporangia. I'll repeat once more, tapetum. It is the innermost layer, 4th layer, and each cell has dense cytoplasm and multinuclei. So, the cells of the tapetum are multinucleated in condition. At maturity, what happens? Pollen sac is formed by the fusion of 2 microsporangia in each lobe. That means what? Each microsporangium is somewhat circular in shape. They join to form a pollen sac. Thus, we say that the cells present in the microsporangium are of similar type and hence they are homogeneous, or we say sporogenous tissue. At maturity, these 2 join to form 2 microsporangia. Both forms 1. On the other hand, these 2 forms 1. Thereby, 2 pollen sacs are formed. Okay. Microsporogenesis. The name itself says microspores are born. Genesis. The process of formation of microspores from pollen mother cell or microspore mother cell by meiosis is called microsporogenesis. That means the cells must be diploid in nature for it to undergo meiosis. Each cell of the sporogenous tissue in a microsporangium acts as a potential, that means active pollen mother cell, that is PMC or microspore mother cell. PMC undergoes meiotic division to form a cluster of 4 cells. This is called microspore tetrad. They will have not yet separated. Pollen grains represents the male gametophyte. We call them as microspores or pollen grains only when the cells separate like this. Pollen grains measures about 25 to 50 microns in diameter. See, microspore mother cell which is diploid undergoes meiosis to form pollen tetrad. Later they get separated to form pollen grains or microspores. Pollen grains are haploid in nature. Structure of pollen grain and is developed into male gametophyte. Each pollen grain has an outer wall which is called exine and the inner wall which is called intine. Outer wall is rough and it is made of a substance called sporopollenin. Okay, it is made of a substance called sporopollenin. Whereas inner wall is smooth and we call that as intine. Followed by this is the plasma membrane which encloses cytoplasm and nucleus. This is again a favorite question for paper setters. Entire wall of pollen grain is said to be sporoderm. It is made of an outer hard layer, exine, and an inner thin wall called intine. This is made of cellulose and pectin. Whereas outer wall is made of, that is exine, is made of sporopollenin. It is related to cutin and suberin. This exine has thin areas which is devoid of sporopollenin. Such regions without sporopollenin are called germ pores. Okay. Yes, students, I told you the meaning of germ pores, that is region devoid of sporopollenin on the exine. Okay. Followed by the entire is a plasma membrane which encloses cytoplasm and nucleus. Sporopollenin is a resistant organic material that can withstand very high temperature as well as strong acids and alkali. There are no enzymes which can degrade sporopollenin. Pollen grains are well preserved as fossils because of the presence of the sporopollenin. Just before the dehiscence of the anther, pollen grain undergoes mitotic division because it's haploid. It has already undergone meiosis, 2N cell, pollen mother cell. So, the pollen grains are haploid. It undergoes mitotic division to form two-cell stage structure. This two-cell stage pollen grain is ready for pollination in 60% of the angiosperms. See, 60% of the angiosperm pollen grain is shed at two-cell stage. Remaining, it is normal. Okay. One of the cell is large vegetative cell and the other is a small generative cell. This vegetative cell has abundant food reserve and it has irregularly shaped nucleus. The other cell, that is a generative cell, has small nucleus which is cell itself is small and the nucleus spindle shaped and it floats in the vegetative cell. Okay. See, in 60% of the angiosperm pollen grains are shed in two-cell stage. In remaining species, generative cell divides mitotically to give rise to two male gametes before the pollen grains are shed. Importance of pollen grains. They are rich in nutrients and thereby they are taken as food supplements. Pollen grains are stored for years together in liquid nitrogen, that is at minus 196 degrees centigrade. Such pollen can be used, okay, as pollen banks in crop breeding program. In the sense they can be used in large amount. Pollen grains of several species cause allergies leading to respiratory disorders like asthma and bronchitis. So, what is the significance of pollen grain? It has both advantages and disadvantages. Pollen grains of the Parthenium or carrot grass or Congress grass. Okay, this came to India as a contaminant with imported wheat and has become ubiquitous in distribution, is found everywhere. In Western countries, pollen products are available in the markets as tablets or syrups. Pollen consumption leads to increase the performance of athletes and race horses.